Um, Thank you once again, everyone, for joining. So this is our Go Parks London and London Friends of Green Spaces Network webinar on citizen science and green and blue spaces. For anyone who maybe is not as familiar with citizen science, it's essentially, I mean, there's actually various definitions of it, but it's essentially science experiments or research done by the public, and often supported by people who are experts, but it contributes to the sort of the wealth of information that we have and that scientists can draw upon. So it's a really helpful thing, and it's also a fun way for the public to learn more about the world around us, and to contribute to our knowledge. So today, I also just want to say I will be doing admin behind the scenes as well, so admitting people and answering questions and stuff. So please bear with me if I suddenly have to focus on that for a second. But so yes, today we are joined by two volunteers at Barnes Common. So we've got Charlotte Tracy, who's going to be speaking to us about the Bioscans project in Barnes Common, and Rachel Walker, who's going to be speaking to us about smart rivers. And then we've got John White from the Friends of Mile End Park, who is going to speak to us about the DNA sampling project that they are doing with the London Natural History Museum. And then we will finish with Janet and John Cadera, who record bird sightings across southeast London in the sort of Rotherhithe and Southwark area. And then at the end, we'll have some time for Q&A. You can pop your questions in the chat along the way, and we can address those as we go, or you can raise your hand at the end of the presentations. So, and also just to say that we're recording. If possible, please mute yourselves if you're not presenting, just so that it's easy for people to hear. And yes, with that, I will hand over to Charlotte and Rachel. Thank you.
Great, thanks very much, Laura. So my name's Rachel Walker, and this evening I'm going to briefly talk to you about Smart Rivers, which is a project I've been volunteering on at Barnes Common Conservation Charity for the last couple of years, and we are based in southwest London. And so Smart Rivers is a citizen science programme that monitors river health. And it's set up by the Wildfish Charity with two main aims. The first, to create a national picture of the state of our rivers to see whether they're able to sustain wildfish populations. And secondly, because there's been quite a decline in Environment Agency sampling over the last couple of years. So it's hoped that this high-level citizen science programme will be able to fill that gap. And the way it works is that we sample freshwater invertebrate species, and then we're able to infer water quality and habitat quality from those results. So we are one of 122 rivers that's part of the Smart Rivers scheme, and we're the only one that's based in London. And we sample along the Beverley Brook, which is a river that runs through Richmond Park all the way to the Thames, and we sample 5 points in a stretch just above Richmond Park. And the reason why Barnes Common wanted to take part and do the Smart Rivers programme is because they were awarded a grant in 2024 to restore a 1.5 kilometre stretch of the Beverley Brook. And as you can see from this photo, there's almost no natural parts of the brook that remain. You can see that the banks have been reinforced, the channel's been straightened and also deepened. And the aim of the project was to restore a more natural meandering flow to the channel, and also to improve habitat diversity for all sorts of species. And you can see here in the photo that a berm's been added to help alter the flow, and also gravel as well, so there's a bit more diversity in substrate. And so the start of the Smart Rivers project was a baseline survey that was done by their team to identify what species we already have. And so the way this is done is we do sampling in spring, autumn every year, and we put on our waders, we get into the river, and we do a kick sweep sample. And this is where you use basically your foot to kick up the sediment at the bottom of the river, and then you catch whatever invertebrates are kind of knocked up with that into a net. We also hand search as well, things like bricks and bottles and other things that might be in the river. And we aim to do a variety of habitats, so along the banks as well, so that we're getting a real kind of representation of the diversity of species there. And so when you first subscribe to Smart Rivers, you'll receive 2 days of training in sample collection and identification. And you also have ongoing support from the wildfish team. So if you find a species or a sample you're unsure about, then they can help you with that identification. And so here you can see us all in our kind of makeshift lab. We are sorting all our samples into major freshwater invertebrate groups using a microscope. There's also the alternative option that once you've collected your sample, you can send it off for professional ID, which is paid for. And so what are we looking for? Well, we're particularly interested in these EPT species, which are mayflies, stoneflies, and caddisflies, because they are particularly sensitive to pollution such as nitrates and phosphates. We don't have stoneflies in Beverley Brook because the habitat isn't correct. So we're particularly interested in any changes to diversity and abundance of mayflies and caddisflies. But we also record other groups as well: beetles, leeches, true flies, mollusks, and various miscellaneous groups. In the top up here, you can see the larvae of damselflies and also crustaceans as well. And we're also particularly interested in looking at the changes in some of these as well, because the true flies, mollusks, and over here on the right, which is a hog lice, which is a crustacean, they are very tolerant to pollution stress, so the opposite of caddisflies and mayflies. So they can live in streams that are heavily polluted, with high levels of sediment and low oxygen levels. So what do our results show so far? Well, if we take a look at the graph, we're comparing the baseline data that was taken before any restoration work was done, that's in brown, and comparing it to our first year set of data, which is in pink. Now, I should caveat by saying this: we only have a first year set of data, and we'll be monitoring these changes over a really long period of time, because the first year of data, that might be those changes we might see might be to do with the weather and climate or differences in predation levels. So we need to sort of see several years of these changes before we're sure of what's going on. But what is interesting to pick out from these results is that you can see that the baseline survey data in brown shows really high abundance of the pollution and stress tolerant species. So the ones that I previously mentioned, the true flies here, huge numbers, mollusks, and also in the crustaceans, those are the hog lice there. But you can also see that has been some change with the first year of post-restoration results. That it's a bit difficult to see down here because we have such huge numbers of the true flies and mollusks, but there has been a significant increase in abundance of the beetles here, leeches, and also the damselfly larvae, and we're hoping that this is a result of the increased habitat diversity in that section of Beverley Brook. But there's also been a significant decrease in the number of true flies here, mollusks, and also worms, and those are the really stress and pollution tolerant ones. We're hoping this is also a good sign that with the change in the kind of flow in Beverley Brook and the habitat differences, that actually there are kind of less places that they can occupy, and that this has been a positive change. But the story with the diversity is different. So again, we're comparing in brown the baseline survey data that was done before restoration work was carried out, and then our first year set of results in pink. And as you can see, there's been no significant change across any of the invertebrate groups that we've measured. And so we have obviously asked ourselves, why might this be? And there are a number of limiting factors that are also at play. So this 1.5 kilometre stretch of Beverley Brook that we've been sampling, it has 9 polluting misconnected outfalls. Now, these are, in our case, domestic properties that have misconnected the waste pipe that comes directly from their toilet or their dishwasher or washing machine, and it's coming out into the surface water pipe directly into Beverley Brook rather than going into kind of the waste pipe and then into the sewer. And you can see here this picture on the right, so I apologize if anybody is eating while they're watching this, but all this grey kind of slime that you can see here, this is a sewage fungus, and that only grows when raw sewage is, you know, quite regularly coming out of this outflow pipe here. So this is literally just pouring out of somebody's toilet straight into Beverley Brook, and we've got 9 of these in this 1.5 kilometre stretch. So you can imagine that the levels of pollution are really high. There's also a couple of other issues that we have to deal with, which is surrounding land use. So obviously I've mentioned there's domestic properties, there's also sports fields on the other side of the bank. So there's likely to be nitrate levels, nitrate pollution, that come off the sports pitches there that flow into the river. And there's obviously pedestrian paths, and there's allotments and all sorts of other things that are contributing to sort of limiting the kind of the benefits of the restoration work that have been done. And the other major issue we have is non-native species. So this is an Asiatic clam, which we first identified last autumn. There's also various other mollusk species in particular that we've discovered, and they're non-native, and they are present in quite significant numbers. And we are unsure of sort of what the sort of, I guess, the consequences of them being there in this habitat. How is it affecting our native species and the habitats they're able to occupy? So there's an awful lot of things kind of going on, contributing to the health of this stretch of river. And so where do we rank with all the smart river hubs that I showed you on that map? Well, a bit depressingly, we are 58 out of 60. So we're very low down in the list of basically the overall quality of the river based on the invertebrates that we have been sampling. And we all joke about this as a team, there's really only one way it can go, and that's for hopefully things to improve. But what it does show you, particularly as we are the only site in London that's doing this, is that, the Beverley Brook is one of the most stressed rivers as part of this scheme, and it really does reflect all of those pressures that I've just mentioned that affect our urban waterways. So there is a huge amount of work that needs to be done. And lastly, I just wanted to share a couple of resources with you if you're interested in finding out more. So if you head over to the Wildfish website under their project section, there's loads of information you can find out there about smart rivers, particularly if you're part of a voluntary group that would like to start something like this where you are. And also you might be familiar with the Biological Recording Company, who do lots of interesting talks. If you Google smart rivers, the Biological Recording Company, you can come up with this talk that was recorded a couple of years ago with Sam Green, who's one of the members of the Wildfish team that we liaise with. So that's it from me. I just want to thank you very much for listening, and I'll be happy to answer any questions that you have after these sessions finish. So I'm hand over to my volunteering partner in crime, Charlotte, and she's going to tell you about another project that we work on.
Hi, I'm Charlotte. I volunteer for Barnes Common also. We're very excited about partnering with Bioscan. We started our partnership with them in September last year, so it's still very new to us. We do have some insights we can share with you, but we've got a long way to go. And I'm just hoping to give you a glimpse into what it involves, how it works, and really our sort of preliminary findings and what impact they might be having for us. So the site in question is actually Barnes Common itself, because the team look after a few sites for the for Richmond Council. The beauty of this project is it's going to provide scientific evidence to allow us to take a deeper dive into the insect ecology at the site, and then this will inform conservation decisions going forward. As I say, it's quite a new method of surveying for us. It's passive collection of insects with the Malaise trap, more on that later. And we're hoping to add to the species list, also look at habitat connectivity with the other sites, and we're also nearby Richmond Park, which is obviously a super interesting site for biodiversity. A side benefit is that the volunteers working on this have already increased their ID knowledge. So Bioscan itself, it's based at the Wellcome Sanger Institute, and it's also in collaboration with the Darwin Tree of Life project. So they've got brilliant resources and are aiming to record 1 million flying insects across the UK. And that's in terms of their biodiversity, and that's over 5 years. So it's going to give a real good baseline and characterise all these insect species that are flying about. They're looking to their partners to provide monthly collections of specimens, which is done by the Malaise trap on site. And then the specimens are actually sent away to be analysed at the Wellcome Sanger Institute. That's using a process called DNA barcoding. It's obviously a complex scientific process, but in a nutshell, the DNA is extracted from a small sample of tissue on the actual organism itself. This is amplified and then sequenced. The end result is something called a barcode, which is a section of the nucleotide sequence, because that can be then matched up against existing barcodes, which are stored on BOLD. It's a database that's open access, and it's also used by scientists. And obviously the idea of this is for scientists and ecologists alike to be able to identify trends and changes over time to the biodiversity of an area such as the UK, and to identify perhaps at-risk species. One of the advantages of this process is it's non-destructive, so the partner sites can get their specimens back for further study. So this map just shows you our project site in the context of the UK. It's an ongoing project, so there's sites being onboarded all the time. I've got a photo here showing.
Sorry, hang on a second, Charlotte. I think are you meant to be sharing your screen?
Yes.
Okay. We can't see anything at the moment, because you mentioned a map, and I thought, okay, wow, that would be interesting.
Oh, thank you. So sorry.
That's
right.
okay.
Can you see anything?
Yeah, see a name.
No,
Can you see
not
it now?
yet. Ah, okay, here we go.
No bother. Okay.
Perfect. Thanks.
Okay, I'm just going to whiz through the slides I've just been talking about, just so it makes a bit more sense. This is a map of Barnes Common, which is the actual location of where we're carrying out the project. And yeah, I've already spoken to you about Bioscan, and this visual might be more meaningful just to think about the DNA barcoding, which obviously I've already spoken about too. Okay, so now you can see the map of the UK with our site on there, and a photograph of our Malaise trap in situ. As you can see, it's in, well, one of the considerations was that it should be in an area of limited public access. Now, being a local nature reserve, it's obviously a lot of public access, and this has to be up for 24 hours each month. So we had to think carefully about placing it. It's in a transitional habitat, so it's actually within some wet woodland, but it's adjacent to a meadow, it's surrounded by scrub, there's a couple of ponds. You can see some dead wood, so we're hoping for some really interesting insect assemblages to come out of that. Placing the trap takes some thought to start with, because you need to think about the insect flight paths and set it up to sort of optimize the success. For those who don't know, a Malaise trap is like a big white tent, and it sort of funnels the insects up who are flying towards the light. So the highest point of the trap, where the bottle is, which is the photograph you see here, is pointing towards the sun. Ours is facing southerly because it needs to be warm and in the sunlight to encourage the insects towards it. I've got some photos here just to give you an insight into the protocol at the site for the field work. It's quite straightforward after you've done it for the first time. You do have to each time look out for spiders. Obviously, the insects would provide a sort of lovely buffet for the spider if you left it in there, and then you wouldn't have anything to collect after 24 hours. So we're always on the lookout for those, but we have actually added some new species to our list as a result. This is at the bench top, so this is in our workspace. It's not the professional lab yet, because as a team, we're responsible for sorting our collected specimens. That's done by size and also morphospecies. It doesn't have to be done exactly because this is all going off to be DNA barcoded, but it just helps to do it systematically, and it's loaded into this 96 well plate, which is designed to work obviously with the laboratory processes at Sanger. There have been some challenges. Bioscan want us to ideally identify it to order levels. That's a taxonomic rank of order. So you're stating whether it's a true fly or a hymenoptera, which is wasps and bees, for example. As you can see from the previous slide, there's some very tiny insects in there. So you do need a microscope, which we did employ after the first session. Setting up last autumn, we've been working through the winter. The wet woodland became more of a pond, so our exact site where we had the malaise actually became submerged. So we just had to shuffle it forward a bit. Every time you set up the trap, you're collecting the metadata, which is then collated with your specimens and passed on. So it's no big deal, but it's just a bit of a hassle to work in the mud. Preliminary data here, because we haven't actually plated up enough specimens to send off to Sanger for analysis. We'll be doing that next month. So just crunching our own numbers, just to give you an idea, the dark green coloured bars on this chart are the Diptera, which is the true fly group. That would be expected in a malaise trap. I mean, it's designed to collect flying insects. But as you can see, as we go towards spring, there's a bit more diversity creeping in. The numbers are also going up happily. Got some photos of some of the more interesting little guys there that we found so far. There's some hoverfly, beetle, and obviously the good old true fly. Once we've made our quarterly submission, we will get report cards back, which is really an interactive device, and it will contain graphics like this lovely sunburst. I've included this because it shows, you know, that the majority of the specimens coming back are indeed Diptera, but it also shows the diversity of the specimens. So what we're hoping to use the results of this project for is obviously long term. So we'll be able to assess the impact of perhaps any changes on site, habitat restoration, and see how that's affecting the numbers of these invertebrates, and also perhaps how it might be related to weather events year on year. And as I said, yeah, provide the scientific evidence to inform our conservation decisions. Here's some resources if you want further information, and look forward to your questions.
Thanks so much, Charlotte. That was really interesting. I've got some questions, but well, I think we'll do the other presentations first, and then come back for questions at the end, if that's all right. But thank you both. That was really interesting. Okay, John. John from Friends of Mowland Park.
Okay. Can you see that okay?
Mm-hmm. Yes, that looks great. Thanks.
Okay, I'm doing it a bit different. I'm doing it as a PDF just because I wasn't too sure how the PowerPoint would work. Yeah, so actually you guys have covered quite a lot of the similar sort of stuff as what I'm going to go on about. So it's an eDNA analysis. We've got a couple of ponds in Mowland Park. What we really are, we usually just do litter picking once a month, and we've tried to sort of move on to other things like bulb planting and looking after woodlands, and we kind of thought, let's have a go at some citizen science. So I just so the one we've been doing is with a Natural History Museum. 650 scientists, well, community scientists, we're not really scientists, took part. It's 362 ponds, and it's mainly about cities. So it's Bristol, Cambridge, London, Manchester, etc. You guys have all already told us what eDNA is really. It's kind of things like shedding skin from various animals. I'll give you one of the things that we didn't detect was terrapins, even though we've got at least 5 of them in our pond. So I think they're not shedding stuff as much as maybe shelled, because it's a hard shell. I don't know. But anyway, so it's things like feces. So I'm sure they do that. It's really about giving you an idea of the biodiversity in the ponds, and we combine it with doing some other stuff, which is like measuring phosphates and nitrates to get some sort of chemical analysis of the pond as well. Okay, so yeah, so you guys have already talked about this really. So we just take a sample, in our case it's pond water, put it in a buffer solution and filter it, send it off, and then the Natural History Museum do the rest in terms of extracting the DNA, doing the PCR analysis and detecting species. And this isn't one of us, but it's this sort of thing. So basically we measure out a certain volume of pond water, put it through this funny little filter thing, then wash it out with a buffer solution to preserve it. That's our pond. That's the Eco pond, available for weddings and stuff. 6,500 a day if you want to come. And there's our heron in the background watching us. Jason, the volunteer, while he's doing it. I think it was Rachel who just explained much better than I ever could about how the science of it works. But we get the results in these little groups. We don't have to do all this complicated sorting under microscopes or anything. I'm quite impressed with that. These are the results across the ponds in the cities. So for instance, 10% of ponds had fox DNA in it. We were one of those. About half had frogs and some snails, earthworms, etc. So that's the general results. They analyze a little this barcode technique, which I knew nothing about. You sort of split them into invertebrates, vertebrates, and stuff like that, and it's done according. So the colours on the graph tell you what it is really. So they put it into each taxonomic group. The blue one is the 12S region, the primer, etc. So we didn't need to know any of this. All we did was prepared the samples and sent them off, and they did the analysis. And these are some of the things, the sort of queries you get at the end of it, which you sometimes, like that picture of the heron that I showed you, we didn't detect the heron at all. And you think, he's there, I'm looking at him. Why? So we had quite a few of those. So there's a few reasons why. Sometimes if the DNA goes into the water and then it just gets gobbled up by something, then you're not going to detect it. We don't actually detect, we don't look at the sediment, so we don't stir up the water. So we're looking at things that are sort of suspended in the water. So that's probably one of the biggest things. So what I'll try and show you, it's still awake. This keeps falling asleep, so I was a bit worried. So this is like one of our ponds, and really what you do is you have a particular ID. You select your pond. I won't go back to that because it takes a little while to load.
Are you showing us a picture? Because we still see the one that says why are animals like expected.
Brilliant.
Okay, great, super. Thanks.
That's worked. Thanks. So look, it does an enormous number of sequences. 67 species were identified. Loads weren't identified, and you can sort of drill down to look into, so 9 vertebrates, etc. So it does it in this sort of starburst way. So you click. Tell me if you can see this. Okay. So you click through. you keep clicking through. It's quite a nice way. So I think you know where this is going to go. Primates, great apes, humans. So that, yeah. So I'll go backwards. I won't. So what else we got? So carnivores. You probably know what we're gonna. So we've got that one, canines. So that's dogs and fox. So it's really good at showing you the things you don't want to see, and you might want to see. You might probably know what the rodent is going to be. So that's a brown rat. So I like it. I mean, it's quite a nice way of looking at things. Then you move down to the invertebrates, and a lot of this is way beyond me. But one of our earlier speakers might be able to see. I don't even know what the seed shrimp is. Seed shrimp is, to be honest. It often comes up with lots of stuff that's unclassified, like it has in this case. So I'm trying to find, if I can find something else for you. So in terms of insects, these things called true flies. You guys have to tell me what a true fly is. Lots of types of midges, biting midges, non-biting midges. Sorry, the contrast is a bit hard to read there, isn't it? So yeah, there's lots and lots of entertainment just looking for various different things. And as you go further down, you end up with just so many more species. So when you get to the bacteria and etc., there's just absolutely an enormous number of species. So you click into that, you just see so many different things. So that's it really. That was a bit of a whistle stop one. But it's a really something it enables you to do is you can look through and look across all the cities and see if the how the nitrate level and the phosphate level is affecting the variety of species or the pH, things like that. So it's quite you can look at your results, but also sort of look at others and maybe learn a bit from them about how comes they've got greater biodiversity than you. So that's it really.
Great.
I don't know.
Thank you so much, John.
Quick enough? Hope so.
It's really interesting to see what can happen in an urban pond as well, because you're in a very busy area, and it's reassuring, I think, to see as well that there's plenty of nature in busy urban areas. Great, thank you. So Janet and John, would you like to tell us about your work? So I don't think this is going to involve any DNA sampling.
Absolutely
this is
not.
more visual identification. So over to you. Thanks.
Hello, Janet and John. We are volunteers at the Stavehill Ecology Park in Rotherhithe, which is run by the conservation volunteers. I started out there in 2016 and then went on to starting recording birds for them. In 2018, we started butterfly transects. For most of the year, we're recording birds for the entire Rotherhithe Peninsula where possible. That includes the remnants of what used to be the Surrey commercial docks. Most of that's disappeared. It was backfilled back when in the 70s, in the late 60s and 70s, when the docks became redundant. What that allowed was the location become quite feral, so to speak, but it provided habitat for lapwing, partridges and little ring plovers. And as the years went by, quite a few species were recorded breeding in Rotherhithe for the first time. Reed warbler bred here for the first time, reed bunting, etc. We had quite a decent reed beds. Those reed beds in our area are fast disappearing. Excuse me. So we try and cover each week at least 2 sites. We have 11 sites that we could potentially cover. That includes Greenland Dock, where we have the last great crested grebes in the Rotherhithe area. We have 1 pair of little grebe which breed in Southwark Park. So we also, apart from the docks, we're covering Southwark Park, and we tend to do that at least 3 times a week where possible. As the season progresses, running into a bit of a lull. The birds have come and it's just nesting stuff. So we monitor what's happening there. During the autumn and the spring, primarily in the autumn migration, there is at Stave Hill an artificial mound, which gives you a fantastic view around Rotherhithe and is excellent for birding, for watching migrating birds through in the autumn. The benefits there are that we see birds over the hill, over the site, which don't land. We've had hawfinches, we've recorded short-eared owls that pass through down through the Lee Valley, brambling, we've had brambling there, etc. So on average, each year, I've gone through my records, we record 77 species of birds through Rotherhithe for the entire peninsula. We also, with the transects, we're recording 24 species.
24 species of butterflies at Stave Hill.
And we've done dragonflies, which is what, 14 species of dragonfly?
We've done 13 species of dragonfly and damselflies and 34 species of moths.
So we do get...
That's just on one site. That's at Stave Hill.
That's the Stave Hill site, yeah. We are trying to do more moth trapping. Part of that, the problem with moth trapping, Stave Hill was public access and all of our sites have public access. So we need to have, there's the shed, which is a fenced off area, which is run by TCV. So we tend to try and set the moth trap there. We then have to go in early, set it up. There are far more experienced people with moth trapping. We're trying to rope them in. We also do bat walks, and we know that we've got at least 6 species of bat within Rotherhithe, including we found brown long-eared bat, and Daubenton's has been logged, but I've never heard them. Hedgehogs, people often ask us about hedgehogs in this area. They were pretty common up until the big redevelopment of the 80s. There was one photographed in Southwark Park about 5 years ago, I think it was. Well, we don't really hear sightings. If we ask people about hedgehogs, they're giving us information that's over 10 years old. So we you know, a lot of the gardens here are paved over. So we have this really amazing wild area in Rotherhithe, which is only 35 years old. So it's quite an interesting place. The whole of Rotherhithe historically has actually recorded something like 150 species, which is quite remarkable. Anything about more stuff? Okay.
How you record?
How we record? Yeah, thanks, reminder. And what are we doing with all of this information? So the birds, I use Bird Track. That's through the British Trust for Ornithology. I upload that every day. We're generally uploading something, at least 3 or 4 records. I then compile that for Rebecca Clark, who runs the Stave Hill Ecology Park. The Ecology Park, sorry, but just to mention it, is backed on by the Globe Pond Nature Reserve, which is then surrounded by Rushadock woodland. So it's a bit of a mishmash. So I tend to record everything within that area, and our butterfly transects also cover that. And we also do work in association with the Southwark Park Association as well, and they get information from us for whatever birds we find. They're more interested in the birds. And all of that, at the end of the year, I try and now combine it into a local bird report, and I hand that over to Rebecca mostly and a few other people. And I think that's pretty much it.
eBird you can do.
You can do. People use eBird. I don't. I find it a bit too complicated for myself. But Bird Track is what I like. I try and collect as much information as possible, including mammals where possible. That includes seals, 2 species of seal, the odd whale that comes up the river, anything. It's quite surprising what happens in this little bit of London, really. And I think that pretty much covers our entire area.
Thank you so
So thank
much.
you.
Sounds really interesting. I'm impressed by, it sounds like you devote quite a lot of time to this. So yeah, well done. Thank you very much. Right, so I think I can open it up to questions now. Does anyone have any questions? I think maybe use the icons to raise your hand or put something in the chat. Or if you wave on your screen, I will try to catch you as well. But I think we've got a few screens going, so that might be a little tricky.
I've got a quick one.
Yeah, go for it.
I wondered about people's backgrounds, really. It sounded like there was some real proper scientists in there as well as citizen scientists. Is that true, Rachel and co?
Well, I'm not actually a scientist, basically, sort of, well, really anymore. I used to work in research and I stopped about 10 years ago, run my own business now. So I'm just doing this as volunteering. So the training is a bit rusty, but certainly like doing all of these projects is kind of, just kind of refreshing all my skills, really, I'd say.
And I'm really in training still because I came to science after I had my children, so more recently, and I started with the Open University to do a biology degree, but then inverts caught my attention, and I've got involved through volunteering really, and it's sort of piqued my interest even more. So yeah. Some science
My
background.
background was so long ago, I can't remember any of the science for it to be useful. It's like, but yeah.
I think volunteering can often be a really interesting way of discovering, one's passion. So I think that's really lovely to hear as well. And John from Rotherhithe, it sounds, to distinguish from John from Myland Park, it sounds like you must have you and Janet must have quite advanced species detection skills, because I know that when I look in my own garden, I can find it quite difficult to work out what's what. So what's your background in this?
I'm an amateur naturalist. I've been interested in natural history since I was a kid. So, and I've always bird watched. And once I got to the point where I was doing part-time work, I then started volunteering at Stave Hill. I got a bit more interested in what was happening with the birds in this area. I started finding old records, and it just turned into quite an interesting area. And I'm fascinated by it now. I grew up with the docks dead. They were a bit of a desolation, really. There were bits I never went to because they were just barren. But I didn't know when I was a kid that there was just so much to see in this area. So I kind of regret that, not seeing little ringed plovers breeding on our doorstep. But we've found enough. We've had yellow-browed warbler here 2 years running. That's a rare warbler that drifts across from Asia into the British Isles in the autumn. We've had a great horned owl that raised a bit of a stir. There was an escaped great horned owl here a couple of years ago that caused chaos. So it's and we've got really good friends who are very knowledgeable, and a friend of ours encouraged us to start vismigging, watching for birds on migration from the top of Stave Hill, and it gets incredibly cold up there. But it's really rewarding. Our first morning, the first session we ever did, we had a short-eared owl come across from Stratford over the river, and you can see this cloud of crows mobbing it. And as it came over our heads and over Stave Hill, the local crows took over from the crows from the north side of the river. So that was, it can get really exciting sort of thing, but often it's not.
That sounds great. Thank you. So we've had a question in the chat. It's Rachel, how do you manage to do your sampling in a reproducible way to enable comparisons over time?
So for the smart rivers, you have a kind of window of time where you take the sampling, and it's usually May and September each year. So you're trying to kind of do it within the same sort of period of couple of weeks, so that you are capturing that time when particular invertebrates are going through that kind of specific life cycle. So we see a big difference, don't we, in spring, where you see a huge number of true fly samples, and we don't see them in September. And so you're taking those 2 points, in the different seasons. We don't particularly do it on any kind of particular sort of weather or anything like that, like you would a butterfly. But yeah, and then you're obviously averaging kind of the results out for the whole year. It's not really seasonally specific. And obviously the timing of the kick sample, we're doing 3 minutes of that. That's divided up between the different types of substrate at the bottom of Beverly Brook. So you have sand and silt and gravel. So you're doing specific amounts of time depending on the percentage that you've got for those different amounts. So that we're able to standardize that in the same way. Both sides of the bank of the river as well. I think they're 30 seconds each. So that's standardized as well. So all the things that we can control in that respect, we do. And obviously the sites are exactly the same. The 5 sites are exactly the same each time as well.
Great, thank you. Art, it looks like you've put your hand up.
Yeah, hi, thanks. Yeah, I just wanted to sort of put a shout out for iNaturalist, and I know that there are other platforms out there, but iNaturalist has been the way that I have managed to get Hampton Heath friends recording stuff for the site. And, we've recorded 607 different species over the last 3 or 4 years, and that's everything from lizards and sort of stuff which is visible, crows and boring things like that, down to we had one fella who came out with a stick and started beating at bushes in order to get little tiny beetles and invertebrates and stuff out of the bushes. So just a big shout out to iNaturalist, really, because it's just such an easy way to, you just take a photograph. It makes a guess as to what it is you're looking at. You can check that, you see if you agree with it. And more to the point, you actually get experts coming in and checking what you've then put forward on your assessment as well. So just look at it. It's a great way in, basically.
Yeah.
And from there,
sorry, go ahead.
we've discovered just on our one scrappy little site, it's only 5 hectare site, we reckon we've found 15 different species of grassland fungi, which could make us, these are something called chegged, CHEGD, which again stands for the different genera involved. And we reckon that puts us on the verge of having our site declared as a metropolitan site of interest rather than just a borough one. So these things can really make a difference. So we've now got eDNA tests sent out at the moment. We managed to get some funding from the Crane Valley Partnership, and so they're out for analysis right now. We'll see what comes back.
Wow, that's incredible. That's a real testament to how important this sort of stuff is, and I hope that you're able to get that that classification as a result of your efforts. That sounds really good. And also, just to say, as you said, it's something that anyone can do. You don't need to be part of a group. You can just go out into your park and take your phone and take some pictures, and then, using the app, it's pretty easy to do. So yeah, good idea, Art. Thank you. Does anyone else have any questions? You can just speak up as well in case I can't see you. Yes, Sean.
Hi, yes, I'd like to endorse the iNaturalist, but the question I have is that we've got a number of people in quite a big park who use Merlin to identify birds and that sort of thing, and I really want to try and get the wider community input into this sort of science, citizen science project, and I wonder if anyone's got ideas as to how we could do that, where we don't have access to great scientific background, but we have a lot of people who are interested in the birds, the flowers, everything in the park. And we've got a lot of people who are interested in the birds, but we want to try and develop it. And has anybody got any ideas as to how we as a community could do that? Yeah.
Yeah. Yeah, just a moment on Merlin. I use Merlin, and I think it's really great. The Merlin is also, you can have some very interesting results with Merlin. It doesn't always translate what it's listening to accurately. So I try and encourage people to look and listen for birds and understand what they've got locally. Southwark Park is not moorland. Last week I got willow ptarmigan, which is not even native to the British Isles. I don't know what it was, but it's great fun. I rate all of these bizarre birds as Merlin megas, and we do get, on average, we can get 1 or 2 every time you go out. The other one that we use PlantNet as well, which is a really nice app. That's pretty accurate. What is it? As iNaturalist, I record for butterflies.
You call butterflies. You can do that one, and all that information goes to the butterfly conservation. So it all goes into the annual reports, and it all comes up as a grid reference, so they know where the butterflies are that you're reporting.
Yeah, I think I've also had some pretty strange. I live in Southwest London, and Merlin recorded for me an oak titmouse, which apparently is native to California. So I was really trying to figure out where that one came from.
Yeah.
Quite a long
Have you
migration.
guys got the right database downloaded?
Everyone said, "Oh, could it detect that you're like American?" And I was like,
Yeah.
"No, that's not what happened." But
Well,
yeah, no. So I think that yeah, the seeing them is also really important. But then if you're dealing with members of the public who are interested but maybe not experienced, I think there's like various places where you can go for courses. I'm trying to remember because I nearly signed up for one myself the other day, and it might have been through the BTO. But John, maybe you can recommend actually some good places for, inexperienced people for bird sighting, knowledge.
The BTO do run identification classes, where you just go onto their website and have a look around. If you within the London area, one of the finest places to bird in the London area for us is Rainham RSPB, and they do guided walks. The guys there who run those walks are fantastic. We know them fairly well. They're really knowledgeable. They'll take out a small group, and because Rainham has got such a varied habitat, you get to grips with waders as well, and brilliant place for waterfowl in winter and spring. I think Barnes Common Wetland Centre is another one people can go to and get experience, just in your local park as well. A good field guide. I always recommend getting a field guide, because you can't go wrong. Well, you can, but it narrows the margins a little bit.
If you want to go onto guided walks, then the London Bird Club does a lot of local walks, and they're part of the London Natural History Society. So you don't have to be a member to go on their walks.
And they do plant and insect
Yeah, they do everything.
walks as well. So they're really good.
Yeah.
Right, thank you.
Sorry, can I say here?
Oh, sorry. I think I'm just going to mute someone here. Okay, go ahead, Sean.
Yeah, thank you for those comments. I don't think it addresses my key question, which I probably didn't put as well as I should have done, which was that I've got a number of people who are interested, vaguely, but it's trying to, if anyone's got experience of how to turn this individual interest and into a sort of group community science activity in a park. Has anyone got any suggestions? I know about the technology. I know about the everything iNaturalist and the PlantNet and that sort of thing. But has anyone experience of getting the group going, and any suggestions as to how we could actually achieve that and sort of practical steps? For I'm because that's the point I'm at the moment. We've got some excellent birders who can tell one little brown job from another, and but we need this community movement and facility to try and develop it. Now, I wonder if anyone's got any experience of that and how it can be developed.
It looks like Art and John have both got some suggestions there. Art, would you like to go first?
Sure. Yeah. Again, just on iNaturalist, there is a facility where you can create a project. So you can create a site boundary. So I've identified the site of interest for nature conservation, which our site is a part of. And then everything which gets recorded, no matter by whom it is, goes into that project. So that's a great way of collecting the stuff. But then what I also have is a weekly newsletter. So anything that gets recorded, which is slightly out of the ordinary, will go into that newsletter. And that just keeps people informed as to what there is at any given time. And it broadens their horizons as well, because I'm not just looking at birds or, I'm looking at damselflies and dragonflies or spangled water beetles or, and some of this stuff is red data listed, and the more red data listed stuff you can find, the more interest you'll get as well, because people, we've got something rare in our park. But so you can set up these projects, you can then communicate with the newsletter, and then the other thing which we've done is got local scout groups involved in doing bio blitzes and that sort of thing. So you actually create an event, and if you can get some involvement from an already organised group of kids, then half the work is done for you already.
Great, thank you.
Oh, sorry,
John.
One
go
more.
ahead.
Yeah, one more thing is doing something slightly unusual. Like a couple of weeks ago, we borrowed an infrared torch, and we went, sorry, not infrared, ultraviolet, and we went out at night looking for caterpillars and stuff. Didn't find any, but we did find some other interesting stuff. And we took some great photographs of plants glowing bright red instead of green, and lichens which are bright orange instead of grey. So you can stick that sort of thing in the newsletter, and again, that piques their interest, and bat detectors and all of those things as well. Make a little event out of just going out with a bat detector.
Great, John White.
Just on a small level, I mean, yeah, all those things are great. We just have a WhatsApp group that people share, maybe a screenshot if they've. I absolutely love Merlin. Janet and John are going to probably shoot me now, but yeah,
we love it. We love it. Don't get us wrong.
So I literally bought this. I bought this to put around my neck. Switch Merlin on, go for a walk. At the end of it, after 5 minutes, you think, "Wow, I've got 13 species of I even got dodo the other day." No, it's not that bad, but it's brilliant. So and you do, and so often I just take a screenshot and send that around the WhatsApp group, and people go, "Oh yeah," And we've also put just a simple laminated poster up next to we've got some swans at the moment. They've just had cygnets 2 weeks ago yesterday, and that's pulling people into our group as well, and people share their photos. Okay, it's just cygnets, etc. But people love the day-to-day soap opera of that. There were 7 born, one died, then another egg the next day hatched, and so that's pulled quite a few people in. But it is and obviously hashtags from Instagram and Twitter. There's a guy who's East London Kingfisher, and he's loads of people follow him, and just he seems to turn up at a place and he sees birds that no one else can, and he gets really good photos. So so there's a little community. But I forgot about the iNaturalist sort of ring fencing. I forgot about that, and that's probably the easiest thing to do, isn't it? Just turn your park into a sort of ring fenced area that people report in. I like that one a lot. Thanks.
Thank you.
Does that help, Sean?
Yeah, it's a big leap between the individuals spotting, and I like the ring fence. I didn't know about. we've got posters we've used. I put stuff up on Facebook of like plants of the week, that sort of thing. And I've got to map out, or try and help map out, a route that is going to involve more and more people in more and more observations and with a purpose. And very few of us have the scientific know-how and background to do this, although there are some. But I'm just interested in other people's experience of how to develop this.
Rachel, it looks like you've got your hand up.
Oh, hi, it's Charlotte here. Just another thought is about getting individuals involved, something like the Bumblebee Conservation Trust, Run the Bee Walk, and there's obviously the UK Butterfly Monitoring Scheme. I think they're like really accessible recording transects that citizens can get involved in. And then if you're doing it on your site, then it ties them into a kind of regular thing. And you know, it might create a sense of community over time if they're walking this transet, you know, during the season, year on year. And it's just a little thought to maybe pique people's interest and get them involved, because obviously the larger invertebrates and things like butterflies, it can be done, you know, while they're mobile, and also with the same with bumblebees. And obviously there's stuff to learn to start to ID them, but it's something you can get your eye into fairly quickly.
Could I make Laura? Could I make a little plug for Giggle? Because
Sure.
we've I ought to introduce myself. I'm from Chislehurst Commons. We've got quite a big friends group and a lot of active people. Nobody wants to do the surveying work, and it falls down to one or two people. We're very, very fortunate to have a couple of scientists within the group. And they become little cells, and we find that the volunteers who have more interest join the particular activities. All the things you've said tonight are brilliant, but one little problem we had early on was that we were being pushed by our borough to enter as much of our data into Giggle, which is the London Record Office, because it was so important. It was linked to the sinks, the local ones and the metropolitan ones. But we found we couldn't get any reports back from them at that point in time. And in fact, we had a subgroup of this group, actually. But out of that, we ended up using iRecord most of the time, because it goes through a process of being verified by somebody who does know more about it from the photograph and the information that we send in. But the really good news is that they now have agreed that we can see the records for our site. So whether it's the records we put on or anybody on the 180 acres that records, we will get the records back. And also we've learnt, as a part through this process, we found lots of groups that weren't sending their records to Giggle. And iRecord now, there's an agreement between Giggle and iRecord. So we know that we send stuff to Giggle, but if it gets missed, it's guaranteed to get through with iRecord. Because I do think it's terribly important, because most of our sites are becoming, well, our site's certainly becoming more biodiverse, because we're much more aware of the habitats that we're trying to create, and we are recording. But thank you very much for this seminar, because it really shows that we're all on a road and we're all making our own way. But I do think it's little steps, and I don't think it's easy. You can't convert a group of volunteers into record keepers if they don't want to do it. But however, you can create little cells within your group who get enthusiastic. So I hope that was some contribution. But Giggle, I think we should be publicising, because it is the London Record Office. So anything that we find should end up on Giggle.
Yes, that's a really good idea. Thank you, Peter. And yeah, I mean, initially we marketed this event as for friends groups only, but actually thought that it was better to just open it up to the public at large, because people, if they have an interest in recording or any of this kind of gathering of information, then we would like them to find a way of getting involved, perhaps contacting a local group and participating. So just trying to seize anyone who might be interested in any form of species recording or any other form of citizen science. So I hope we have gone some way towards achieving that. I'll wrap things up now because we've gone a little bit over time, but I just want to really thank our speakers. I found that really, really interesting. I have some questions actually, so I might follow up with some of you afterwards. But just to say also thank you for all of you for joining. And yeah, we had a lot of interest in this session. I will be sending out a link to the recording afterwards. And yes, just thank you everyone, and I hope you enjoy your evenings.